I'm going to get started. Welcome, everyone. I'm so thrilled you are all here with us for our inaugural virtual Bite of Science. This is our first attempt at doing Bite of Science virtual. So I'm very thankful for all of our teachers who've joined us today, as well as our two presenters. So I'm thrilled we can all try to do this. I say it's inaugural just to bear with us as we figure out, technology stuff. Today we'll be hearing from Dr. Maren Roman, who is a professor over at Virginia Tech in the Department of Sustainable Biomaterials. And with that, I will hand it over to Maren. So welcome.
So hopefully you can see the slides now. And so as Kim said, I'm currently an associate professor in the Department of Sustainable Biomaterials. And I'm posting my email address here, maren.roman@vt.edu. So if you have any questions or would like to contact me after my presentation about any of the opportunities or tools that I'm talking about, feel free to just send me an email. As Kim mentioned, I've recently become the director of a $23 million project funded by the National Science Foundation. And today I'll be sharing a little bit about my life journey with you and how I got to where I am today. So I grew up in Germany, in the second largest city of Germany, which is Hamburg. Hamburg is right where the green dot is, so it's in the north of Germany. Hamburg is very similar to Boston, except larger. It's about 2.6 times the size of Boston. It has two large rivers running through it, and it has a large harbor as well. So during high school, I was rowing crew, that's it. And I got around town on my bicycle. So that's what we did as teenagers. We were riding our bikes, sometimes up to 45 minutes at a time to get across town. In the last couple of years in high school, we were choosing concentrations, and my concentrations were math and biology. But it was really the chemistry teacher who hit the spot with me. So I got really curious about at the molecular level. And so I went to Clausthal University of Technology to study chemistry. And that is located right about here in the center of Germany. And it's a mountainous area. The town was very small. And it was quite a culture shock initially to move from a large city with 1.8 million people to a small town that has just about 20,000 people. And but then I learned to appreciate nature, and I really started to enjoy cross-country skiing in the forests and riding my mountain bike and jogging and all these things through the nature. So I started enjoying and appreciating nature. At that time, we didn't have anything that compares to a bachelor degree. So my first degree is more comparable to a master's degree. It's 4 years of coursework and 1 year of research with a thesis at the end. So I got a master's degree, or comparable to a master's degree, in chemistry. And during my studies, I also had the opportunity to study abroad. I was lucky to get funded 3 times by the Erasmus program. That's a program in the European Union that supports students going to other states, to other countries in the European Union. So my first trip was a 5-month trip to Padua in Italy, which is only 30 minutes by train from Venice. So I got to see Venice in the off-peak season, and I was fluent in Italian by the time I got back home. My second trip was to Aberdeen in Scotland, and that was about 3 months long. And I got, that was really good for my English. It was, of course, a challenge to understand the Scottish accent, but I had a good time, and it was a big help to get fluent in English. And then the third trip was just a 2-week trip to France, right here near Luxembourg. And that was a 2-week language course in French. And because I had chosen Latin during high school, that was my first exposure to the French language. And then at the end of my master's studies, I was asked by my advisor whether I would be interested in coming to the United States and doing some doctoral research in a lab of a friend of his that was looking for a PhD student. So I came to the United States in 1997, and the initial plan was just to do 2 or 3 years of research and then return to Germany and finish my doctorate degree over there. But somehow that didn't work out. As you can see, I'm still here. It might have had something to do with me meeting my future husband at the university, as it happens so often. But I do enjoy living here in the United States. So my first stop was Syracuse, New York. I went to get my doctoral degree in the College of Environmental Science and Forestry. I really learned the term lake effect at that time. So I experienced some of the coldest winters and the most snow that I had ever experienced in my life. And I got through it 5.5 years in Syracuse, and I got my PhD in chemistry, in polymer chemistry. And after that, I moved further north to Montreal, and I did 2 years of postdoctoral studies at the Department of Chemistry at McGill University. If Syracuse wasn't cold enough, Montreal was colder in the winter, and they had so much snow that they actually had to move the snow out of the city on trucks because they just didn't have enough room in the city to accommodate all the snow. So I did 2 years of postdoctoral studies in Montreal, and in the summer between the first year and the second year, I had an opportunity to do a summer internship at Eastman Chemical Company in Kingsport, Tennessee. So that's right down here in Tennessee. And the climate there was more comparable to what I had experienced in Italy. So it was, of course, very hot in the summer, but I did appreciate the experience of doing some research in an industrial setting. So that was very different from the academic setting. I spent, I think, about 3 months in Kingsport, Tennessee, and then I went back to Montreal to finish my postdoctoral studies. And after 2 years, there was an opportunity for a faculty position at Virginia Tech. So I interviewed and I got the position. And so since 2004, I have been in Blacksburg, Virginia, at Virginia Tech. And I have to say that climate fits me pretty well. It's not too hot because we're in the mountain, slightly elevated here in Blacksburg. And the winters, you do get snow occasionally, but most of the time it's relatively mild. So when we look at the scientific journey, my first research experience was something comparable to a senior design project. In the 4th year of my undergraduate studies, we had to do a 6-week research project, and I chose organic chemistry. And my task was to synthesize a molecule that was a frog toxin. And so I was given the molecule, and I had an advisor that was giving me different strategies to synthesize the molecule. And I started setting up the apparatus and putting all the chemicals in, and the first strategy didn't work out, the second strategy didn't work out. So I kept repeating experiments, and I think by the end of my 6 weeks, I had done probably 6 or more different attempts, and none of them worked out. So I had to write a report describing all the experiments that I did, and I couldn't show for it. I didn't have any product to show for it. So that was very frustrating. So I decided that OChem is not for me. So I ended up doing my master's research in physical chemistry instead. And at that time, we had a couple of different areas of physical chemistry. I chose to focus in liquid crystals. And up here we have some images. If you're not familiar with liquid crystals, those are materials that have a state of matter that is between a totally liquid state where the material is fluid and has no odor. So here we have some liquid state where, in this case, these elongated molecules are completely randomly oriented. And in the solid state, there's a very rigid ordered structure, which is the crystal lattice. And these liquid crystalline materials, they have a state in between where they are fluid, like in a liquid, but they have some degree of order of the molecules. So what I studied was these mixtures of benzoic acids. So we have two different molecules. Here's one type of molecule and here's the other type of molecule, and I would mix them together, and when they form these hydrogen bonds here between the hydroxyl group and the oxygen, you get these very long molecules or structures here that would be these rods here, and I would observe these different liquid crystalline phases. So on the right here, we see an image of a so-called blue phase, and I was able to publish that. That was only the second blue phase observed in the literature for such systems. Then I came to Syracuse, New York, and did my PhD research on polymers and cellulose nanocrystals. And I will tell you a little bit more about cellulose nanocrystals in a moment. So these are tiny particles made of cellulose. And my research and my PhD research used a polymer and mixing the cellulose nanocrystals in with the polymer in order to stiffen the polymer. My third project was the postdoctoral research, where I combined the liquid crystalline properties and the cellulose nanocrystals and studied these liquid crystalline films. So here we have a film that has some ordered structures, and I was able to model the structure that is present in these films. So what are cellulose nanocrystals? These are cellulose itself is a string of glucose molecules. So here's a string, and in nature, these molecules aggregate to form crystalline fibrils, which are shown here. And these fibrils are what makes trees strong. So here you can see that these fibrils are making up the cell wall of these wood cells. Here you see the structure of wood. And on the right-hand side, we see an atomic force microscope image of the surface of a wood fiber, and you see these fibrils there. We can treat these fibrils with acids, so we can use either hydrochloric acid or sulfuric acid, and that breaks these fibrils down, and we end up with these rod-like structures, which are the cellulose nanocrystals. They are very small. On this image here, you see this is a human hair, which has a diameter of about 50 micrometers. And so the image here is 2.5 micrometers across. So that gives you an idea of the size of these particles. So I started at Virginia Tech in 2004. My research since then focused mostly on the properties of cellulose nanocrystals and some potential applications of these particles. So up at the top, we see a suspension of cellulose nanocrystals in water, and at a concentration of 10 weight%, we see a phase separation. So we see that we have a phase at the top that's milky and a phase at the bottom. And the bottom phase has these iridescent colors. And you can also dry that into an iridescent film. And this is a liquid crystal of these cellulose nanocrystals. So those are some of the properties that we study and the potential applications. In one case, we mix the cellulose nanocrystals with another material called chitosan and form these microparticles. And we had a project funded by the National Science Foundation to investigate whether these could be used to deliver drugs for oral delivery, so that would be taken by mouth. In another project, we modified the surface of the cellulose nanocrystals. Here's a fluorescent marker. On the other side, we attached a folic acid molecule, and then these molecules are taken up by cancer cells. So we did some animal experiments where we injected these materials into the tail vein of the mouse, and the mice had tumors on the inside here near in their tummies, basically. And what we see is that in a higher concentration, 4 hours after injection, the materials are accumulating in the tumors, and that means that we can use these materials to deliver cancer drugs directly to the tumor, which would then spare the healthy tissues from the detrimental effects of the cancer drugs. Another project, we used the cellulose nanocrystals to mix it with some polymer, and we used 3D printing to print these kinds of scaffolds that could be used to heal bone defects. And now coming to how did I become a director of a $23 million project. I basically used a couple of different experiences from my past. I served for 2 years as the program chair of the Cellulose and Renewable Materials Division of the American Chemical Society. And during that time, I made a lot of connections with other scientists all over the US and even internationally. So that was one of the things that contributed to my success. Another one was in 2008, I was invited to serve on a review panel for a $100 million Department of Energy Research Center. And that gave me some experience with a large research center. So then in November of 2018, NSF came out with a solicitation for materials innovation platforms, and I led an effort, a proposal to that program, and these are a couple. The other additional factors that were important are a passion for the project, a vision for a large project, and then a bit of boldness in approaching some high-profile scientists and just asking them to participate, and a bit of naivete, not in order to attempt some large project like that. The project that got funded. Let me back up for a moment. This is a large center that focuses on glycomaterials. And the project is called Glyco MIP, that stands for Materials Innovation Platform. And I wanted to tell you briefly about what glycomaterials are. Everybody has probably heard of carbohydrates in the context of nutrition facts, but there's much more to carbohydrates than nutrition facts. And so that's what my title of the presentation was. So carbohydrates, they are hydrates of carbon. They have carbon in them, and they have H2O. They form these ring structures here, and when we have a 6-membered ring, this is called a pyranose. Sometimes they form these 5-membered rings, which is called a furanose, and they are everywhere in nature. We eat them. They are in food. They are in plants, as we have seen, and they are also in humans and every living organism. They are present. So let me show you a resource, and that is called a carbohydrate builder. So if you ever talk about carbohydrates in your classrooms, maybe you have maybe you're talking about lactose in the context of lactose intolerance. Let me share a different screen here with you quickly. So here's a tool called Glycam on the website called glycam.org. And they have a carbohydrate builder here. And here you can assemble carbohydrate molecules. So for example, if you are looking at what the molecule lactose looks like, it's called beta-D-galactopyranosyl 1,4-D glucose. So you can look up what this molecule is like. And then you go to the carbohydrate builder and you choose galactose is GAL. We just saw that it has the linkage beta. Let me go back here. 1,4, so you choose 1,4 over here, and then it says glucose. So then you choose GLC, which is the abbreviation for glucose, and then you attach a hydroxyl group and you press done, and then you go to continue, and it will build that structure for you. So when you go to visualize here, then you have this molecule and you can rotate it, you can download it, you can measure bond angles when you double click on one atom and you double click on another. This measures the length, measure the bond angles, bond length between two atoms, and you can also measure bond angles. So when you go from here to another atom and then another atom, here you can measure the angle in between these two bonds. So this is a tool where you could build complicated carbohydrates and then use that in your classroom. You can download the molecule, and then you can use a free viewer to share that molecule with your students. They can use free viewers to study this molecule. And this is one of the tools that I wanted to share with you. So I talked about the Glycam tool. We have at Virginia Tech, the Fralin Life Sciences Institute offers the Biotech in a Box program. Those are free, those are kits that are being shipped to high schools and community college classrooms. Virginia Tech pays the round trip shipping, and those have activities in them that you can use in your classroom. And then a couple of programs I wanted to mention are, we have again, the Fralin Life Science Institute has Kids Tech University, the program, some class tours, and some research experience for high schoolers. And then we also have programs in the College of Engineering. One is called Pathways for Future Engineers. And then the College of Natural Resources and Environment has a program, a 3-day summer camp that's called Inside Trees. So if you would like to learn more about some of these programs, feel free to send me an email. And I will stop here so that you have some time to ask me questions.
Oh, thank you, Maren. So I guess we can open the floor to questions. So if anyone has a question, I guess you can unmute yourself and ask it. If people want to think for a while. I had a question. I wanted to hear more about your the bone scaffolding.
Okay. So the thing is that when you break a bone, if it's just a small fracture, it can heal naturally. But if it is a very large fracture, then the bone needs a little bit of help. And so there's something called bone tissue engineering, where there are materials that can be inserted into the gap that will help the bone to sort of regenerate itself. And these materials can be either natural, so they can take, for example, a piece of bone from a place in your hip where you don't need it, and they can put that in. Those have some disadvantages. So there's a lot of research on looking into synthetic materials to do that. And currently there's a lot of interest in 3D printing, because what you can do is you can use a CAT scan to determine the shape of the piece that is needed. When you're thinking about, for example, if somebody has a damage to their head, you want a custom shape. So then you can do a CAT scan to determine what's shape that material needs to be, and then you can use 3D printing to print something specifically for that patient.
Awesome, thank you. Are there any other questions?
If there are no questions, I can cover some of the slides that I skipped.
Please do.
All right. Okay, so I wanted to talk just a little bit about small carbohydrates. So I said that glycomaterials are the topic of glycomip. And the definition of glycomaterials is that those are materials that are composed of carbohydrates, or that contain carbohydrates. So carbohydrates are synthesized in plants by photosynthesis, and that's I don't know if it's taught in middle school. So I know that you know that. I just wanted to bring that up here because it puts this in context. So the carbohydrates are synthesized in the plant using sunlight, carbon dioxide, and water. And you're getting, oops, let me just get my pen back here. So we have glucose is formed in that process, and then glucose can be converted into other carbohydrates using a process called sugar interconversion. Now these carbohydrates are then either used to make the plant. So we saw that carbohydrates are used in as structural materials in wood, for example, but they can also be stored for energy inside certain parts of the plant, and then we're eating those plants for energy. So we get the energy out in one of several ways, either through the process of respiration. So when we breathe oxygen in, we eat the carbohydrates, then so here we are taking oxygen in, we're eating the carbohydrates, and in the process of respiration, we're generating carbon dioxide, we're generating water, and we're getting energy from it. A similar process happens with bacteria. They can also do this conversion, and the same happens if we're burning wood. So that's one of the concepts that I wanted to cover. And the next slide, here I wanted to introduce a little bit of carbohydrate chemistry so that you can use that carbohydrate builder a little bit better, because you have in this carbohydrate builder, you have to choose, for example, the linkage between the sugars. And for that, I wanted to cover a little bit here. We have certain way to label the carbon atoms in these sugars. This is always carbon 1, and then we're counting clockwise. So this would be carbon 2, this would be carbon 3, and this is carbon 4. So when you're using the carbohydrate builder, you're specifying, for example, how these sugars are connected. And then on the next slide, I was just trying to show you the different places where carbohydrates and glycomaterials are, where they occur. So the top image here shows the surface of a cell. So this is a cell membrane, and embedded in the cell membrane we have proteins, and we have, let me see where we have lipids. So these molecules with the 2 legs, you're probably familiar with this. This is a phospholipid, this is the hydrophobic part, and this is the phosphor group head. And we have these carbohydrate chains attached to these molecules. So we have glycolipids, or carbohydrates attached to lipids, and we also have glycoproteins. I'm looking for there. Glycoproteins is carbohydrates attached to proteins. So glycomaterials are occurring in every single organism. at the cellular level and at, also at a larger level. So for example, between the cells in the extracellular matrix, we also have glycomaterials present. So the center that was funded, the purpose of the center is to support research on glycomaterials and to support the development of new materials based on carbohydrates.
All right, well, thank you. Thank you, Summer. Sorry, Summer just added just a comment. I think some of my students would benefit from the carbohydrate builder when I cover macromolecules.
Okay, yeah, and if you have questions, I mean, this was very short, so feel free to send me an email and we can set up another meeting and we can go through, you know, some more specific needs that you have.
All right, thank you so much, Dr. Maren Roman.